as we mentioned, DNA replication is not exactly perfect. We do have to go through DNA repair mechanisms. There are 3 types. Proofreading, this goes on during replication. So this is like spell check on your computer from the standpoint that it checks as you write. So, you know, when you spell a word wrong, it underlines it in red. So this is during the DNA rep process. Mismatch and excision repair are after polymerase has finished, and it's going back and checking again. Now, DNA repair mechanisms are critical because DNA polymerase makes mistakes big time. There's an error rate of about 1 in 10 to the 4th with just DNA polymerase. But if you add in the repair mechanisms, you can get that error rate down to 1 in 10 to the 9th. So when we look at these repair types, this is just a kind of diagram for you how they're occurring. The polymerase is going to find the problem, kick out the bad nucleotide, and put in the proper one. Because remember, in this, as this is going on inside your nucleus, there are just nucleotides bouncing all over the place. The reason they sit down in the place where they belong or where they should be to match up is because of chemical structure. So it's all 3D chemical structure again. And occasionally it goes wrong. But DNA polymerase hopefully finds that. And you see it's indicated as kind of this donut that's running along the DNA strand. That's accurate. Why would we want this? Because if there's a problem, if something is out of place, it actually sticks out on the DNA strand many times. And so what we end up with is a lump in our DNA strand that polymerase, if it's a donut riding along, can't get past. So it knows that there's an error there that has to be fixed. DNA repair, of course, is extremely costly. This is a high-energy process, lots of things involved, lots of things going on, plus it slows down the whole DNA replication process. But without it, we would build up so many DNA mistakes that we would actually die. The cells would not survive it. So, guesses. How many mutations are actually created in each cell division? So this would be per 24 hours if your DNA repair system is functioning. Guesses? 1,000? 10,000? 10? About 3. So you get about 3 mistakes every day in your cells, in each one of them, because of DNA repair mistakes. Now, that doesn't mean that you're going to mutate immediately and die. These mistakes are many times what we call silent mutations, meaning that they make absolutely no difference in the functioning of your cells. And we'll talk more about these later. Other sources of DNA damage other than just DNA polymerase screws up. Normal metabolism. We've talked about free radicals. They come in, attack things. They're going to do some damage, some mutation sites. Environmental things like UV rays, smoking, unfortunately burning charcoal and breathing that in, not so good for us. Mitochondrial DNA damage. Sure. Your DNA repair systems have to deal with 50 to 500,000 what we call lesions or mutation sites per day. So not only is your poor DNA repair system trying to deal with the mistakes that polymerase made, now it has to deal with whatever we expose it to. And as we get older, these things build up. So as DNA repair slows down, and we end up with damage essentially exceeding repair, the cells can either go into a state of dormancy, they'll just stop dividing. Your body will recognize that there's something wrong with the cell and actually program it to be killed, something known as apoptosis. This is cellular suicide, essentially. And then we can also end up with cancer states. So remember, cancer is uncontrolled cell division. So we can end up with a state where the cell will just go into this division and divide and divide and divide. And unfortunately, that spreads that mutation. We do have conditions associated with decreased repair mechanisms. Werner syndrome is associated with poor repair. So this DNA repair system is not working properly. And you see this woman at the age of 15 and then again at 48. I think she looks much older than 48. We have accelerated aging associated with this. Now this is different from things like progeria. These are very different syndromes. This one is specifically based on a poor repair mechanism. Xeroderma pigmentosum is another one. XP we see a lot more common recently, and it's also due to poor repair. These folks, if they go outside and are exposed to sunlight, they get immediate sunburns, and it's not just a regular, you're kind of a little pink sunburn. These are third-degree burns essentially immediately. The opposite, of course, is also true. If you can have bad repair, you can also have really, really good repair, right? And we see this in one of our archaea friends, one of our extremophiles, something known as radiodurans. These are amazing. These are an extremophile that are actually being used in bioremediation to clean up gamma radiation because they are extremely good at taking in chemical radiation and dealing with it. So they actually take in very, very high doses of what we call gray units. So 10 kills you. 10 units. Radiodurans can stand up to 15,000 of these units, and 37% of them will survive. That's amazing. They also don't care about heat or cold or dehydration or anything else. That's why they're listed as a polyextremophile. These things really don't care about anything. They're pretty amazing.